Hello everyone, I am Girija, pursuing second MSc Microbiology, Vivekananda Arts and Science College for Women, Veerachpalayam, Sangagiri. This presentation is prepared by under the guidance of Dr. R. Dinesh Kumar, Assistant Professor, Department of Microbiology. Today, I am going to present on systematic acquired resistance. Systematic acquired resistance is a natural defense mechanism in plants. It provides a long-lasting protection against a wide range of pathogens such as bacteria, viruses, and fungi. Systematic acquired resistance is activated by a plant experiences an initial pathogen attack. Once if activated, the resistance spread throughout the plant. So it is a broad spectrum and long lasting and works like a memory system. So the plant's responses faster when attacked again. Now let's see about historical background of systematic acquired resistance. The concept of systematic acquired resistance was first observed in 1961 in tobacco plants infected with the tobacco mosaic virus. Later scientists discovered special proteins, it is called PR proteins, and identified salicylic acid as a key signaling molecule. Today we know about systematic acquired resistance also involve a gene networks, and even we know it is a epigenetic memory. Now we compare the systematic acquired resistance and other defenses. Systematic acquired resistance different from ISR. ISR means induced systematic resistance, which is triggered by beneficial microbes. But systematic acquired resistance mainly depends on salicylic acid, while ISR often involves a jasmonic acid and ethylene pathways. Systematic acquired resistance can be induced by biotropic pathogens like viruses and fungi. Sometimes a necrophytic pathogens by chemical compounds such as salicylic acid and BTH, and even by stresses like UV light or beneficial microbes. Now let's see about the how salicylic acid act as a main regulator. Salicylic acid act as a main regulator of systematic acquired resistance. It activates local defense genes and generates mobile signals, primes distant tissues, and induces PR proteins. At the site of infection, salicylic acid accumulates and activating a key regulator that is called NPR1. This leads to defense mechanism expression. For long distance signaling, molecules such as methylene salicylate, glycerol 3-phosphate, and azelaic acid act as a mobile signals and transported through the plant to activate systematic resistance. What is NPR1? NPR1 is a master transcriptional regulator. It activates the genes like PR1, PR2, PR5 and works together with the transcription factors such as WRKY and TGA. Now we see about PR proteins. What is PR proteins? Pathogens related proteins is called PR proteins. PR proteins are small, stable and antimicrobial proteins that are directly attack pathogens. They are an important output of systematic acquired resistance. Now see the priming in plants. Priming is when plants prepare their defense system without always keeping it active. It means they respond faster and stronger during future infections, saving energy compared to constant defense. The main positive and negative regulators of systematic acquired resistance is NPR1, WRKY70 and EDSI. These regulators are the positive regulators, while NPR3 and NPR4 and some WRKY proteins act as a negative regulators. There is also across talk with other pathways like jasmonic acid and ethylene and abscisic acid. In systematic acquired resistance, the studies in Arabidopsis, tobacco, tomato, rice, wheat, cucumber, and beans show that systematic acquired resistance is highly conserved mechanism across plant species. The environmental factors influencing systematic acquired resistance is a temperature, light, water, and nutrient status influencing systematic acquired resistance. For example, high temperature can reduce the systematic acquired resistance effectiveness, while nitrogen and phosphorus levels can affect the defense gene expression. Now see the agricultural applications in systematic acquired resistance. Chemical inducers like BTH and INA can artificially trigger the systematic acquired resistance. Genetic engineering to enhance the NPR1 or PR proteins can also increase the resistance, and Harpin proteins from bacteria or another option. See the role in sustainable agriculture. Systematic acquired resistance helps to reduce the pesticide use and provide long-lasting protection and support eco-friendly crop management. It is a key component of integrated pest management. The limitations of systematic acquired resistance is, however, systematic acquired resistance has some limitations. It consumes plants energy and may reduce the growth depending on timing and environment, and is less effective against necrophytic pathogens. The current research are going in systematic acquired resistance is, the research is now focusing on epigenetic regulation systems, biological approaches in CRISPR editing, biosensors, and nanotechnology for targeting delivery of systematic acquired resistance inducers. And what are the climatic stresses affecting systematic acquired resistance is, the heat, drought can affect the systematic acquired resistance. Therefore, future strategies aims to breed heat tolerant crops and use stress responsive systematic acquired inducers. Future perspectives. In the future, precision agriculture tools like smart sensors and synthetic biology for engineered systematic acquired circuits and climate resilient strategies will play a big role. Let's move on the conclusion. Systematic acquired resistance is a powerful defense mechanism in plants that provides a durable broad spectrum resistance. Although it has challenges, it offers a great opportunities for sustainable agriculture. Thank you.